Hi, everyone. Thank you so much for joining us today to learn about multiplex PCR, formerly a hot mess and now ridiculously fast, easy, and affordable. Since we're right at the top of the hour, I know there are still some people trickling in. So while we wait for everybody to get settled in, I'd like to throw up a quick poll to learn about how everyone here is or whether they are already doing multiplex experiments. So you should see that on your screen. Yeah, so we're just curious to know whether people are multiplexing, and if so, how much you multiplex. Um, Again, as people come in, I'd like to introduce our speaker for the day. Uh, with me, I have Eileen Schum. She is the founding scientist and VP of product here at Countable Labs. And it's really thanks to her and a couple of other founding scientists that we have this amazing platform that makes multiplexing so easy, really. So thank you all for contributing to the poll, joining us today. Just to kind of give a quick rundown, it looks like we have a real mix of audience today. Some people doing high-plex, some people kind of in the mid realm, and then some people who aren't using it at all. So I really hope that today's webinar helps us learn how multiplexing really doesn't have to be a pain anymore and can be a lot more accessible to everyone. So I will be back at the end to answer some questions. And with that, I will leave you, Aline, to lead our audience in a discussion about multiplex PCR.
All right. Thank you, Stephanie, for the introduction. So, hi, everybody. My name is Aline Shum, and I'm one of the founding scientists here at Countable Labs. Just a bit about myself. I spent over 20 years as a genomic scientist, and most of the time, honestly, I've been running, qPCR, right? Thousands of plates by now. And honestly, like many of you, I first defaulted to SYBR Green for a lot of my analysis. It was quick, it was cheap, and it felt like the path of least resistance. But the truth is, a lot of times it doesn't give us the depth of answers I really wanted. So multiplexing always seems like a good idea, but in practice, it was messy, expensive, and frustrating. Hence, our cheeky title today for the webinar. But obviously, we know the value of multiplexing, right? If you can reliably measure multiple targets in one tube, you get more data from the same sample and you reduce the variability. But obviously, the execution hasn't kept up. So let me just introduce to everybody so that we are level setting here in terms of the value of multiplexing, right? By multiplexing, essentially you're looking at multiple targets in the same reaction. So first and foremost, you get more data. You can look at, say, your wild type or rare targets together, or different targets that you want to normalize against to understand, for example, effective copy number. And the value also for multiplexing is to allow you to be more accurate, right? Fewer pipetting errors. You don't have to compare data from well to well. It reduces your variability. And three, honestly, saves costs because by looking at more with fewer reactions, you spend less not only on your own time and labor, but also reagents and consumables, right? So just giving you guys a quick example here. One of our CLIA customers is doing monitoring of 4 potential mutations per patient. And currently they're doing traditional qPCR where they're doing one reaction per mutation. And then they also would have to do standards, as you know, for qPCR in order to get the accuracy and to understand the mutation burden. But with what I'm going to introduce today with Countable PCR, those mutations can be easily put together in a single tube. And the result really is dramatic because you can have up to 58% or more cost savings with just one single assay. And the difference between multiplexing, right, is not just only cost, but when you imagine needing to run over 1,000 of these patient samples per month, it saves a lot of labor as well. But obviously, there's a lot of value in multiplexing. But when we talk to users like yourself, typically what we see is that multiplexing is more difficult than advertised, right? Just because like an instrument can support this many colors, most of the time, a lot of us just do much fewer plex than that, or even not multiplex at all. And I want to spend a little bit of time dissecting why today, really. The first piece, right, is that multiplexing is actually quite expensive right now. So, um, you have to buy probes and they cost a lot of time and cost a lot of money for you to even receive these probes. So a lot of researchers hesitate to move to multiplexing unless they have the time and resources to be able to do that. And the second piece is that it does take quite a long time to develop. Depending on who I ask, right, it could range from like 6 months to over a year to really develop a multiplex assay that is reliable and that you can run day in and day out, especially as you increase plexy. The complexity of how the PCR is done and how the data is optimized actually it increases sort of the length of time required to do your assay optimization. So a lot of times when you're in the early phase of either in research or discovery, right? Like we tend to settle for less, and I myself am one of those, right? I just kind of rely on SYBR Green to make it like fast, right? It's cost effective, it's easy. I don't have to think about like too much about design and optimization. But the issue is I don't really get like a complete picture of the real biology. And I also know, right, like if I have to put an assay to analytical development or develop the clinical diagnostic assays, eventually I would have to go to multiplexing. So either way, we kind of split the workflow in the sense that we do singleplex in early discovery, but multiplex later. But then we still have to spend that time and the arduous process of optimization to get there. And what we like to kind of talk about today is like how multiplexing is slowing you down today and how and why it takes weeks and months and why it prevents you from launching your products or getting your assays or getting your results ready. And I want to talk about like that it's not you, it's really the PCR platform that you have right now. For example, if you're a digital PCR or a qPCR user, how the technologies right now actually doesn't make multiplexing easy. And plus, adding to TACMan Pro costs adds to sort of your obstacles as well. So in the next section, I want to talk a little bit about like why, what is the fundamental sort of molecular biology behind why multiplexing is so difficult right now. And let's start with, say, qPCR, right? We all know that qPCR is very powerful, and essentially you just basically put all the reaction in the same vessel. Essentially, it's a bulk reaction in the sense. I'm showing you here a schematic of each star is essentially the colored ones is what target you're looking for. So for example, if you're looking at threeplex, you're looking at gene A, gene B, and gene C. What you actually see in a PCR reaction is that all of these targets and all of these starting molecule templates are actually kind of competing with each other for PCR reagents. Think about like they fight for the polymerase, the NTPs, and whatnot. And two, each of these targets actually have different PCR efficiency. So you have to not only fight for those things, but you also often always see that different amplification efficiency or the abundance of targets actually skew your results. And this is the type of PCR bias that you typically see. And it's quite apparent. I'll show you some data of how that impacts your multiplexing later. And I'm sure some of us here are familiar with digital PCR, right? Multiplexing and digital PCR can be more accessible, but it still can take months to optimize. And if I take the same example here and just apply it to like the principles of digital PCR, essentially in digital PCR, you're trying to partition the same reaction into thousands of compartments, right? A lot of times your target could be at a much higher magnitude, and that's actually quite often, right? Like the number of partitions that's available in digital PCR is low. So what you end up getting is essentially a realm where you have overloaded partitions, whereby if you have these stars, you're trying to like do these multiplex assays. Some partitions might have one, so that's great, but some partitions might have multiple. You can see here it could be, 4 or 5. And what you see is because this one, for example, is amplifying by itself, and these are amplifying with competition. You start seeing actually different phenotypes of data that comes out of a digital PCR reaction. Just showing you here in a threeplex, type of signal complexity you can get unless you perfect the optimization of your multiplex assay. What you get is that you get these like maybe in between signals when there is a partition that has two or more targets. And the result is, and this is a data from our collaborator, essentially they're a powerhouse in digital PCR development. And even for them, when they develop a multiplex assay, sometimes even on the best assays, the data can be highly subjective. So you can see here, these in-between phenotypes are actually represented here in this maroon cluster here. They're not as efficient as if it's only single occupant. And this variation in signal change like affects how like we draw thresholds. So we've also talked to customers where like maybe your colleague would draw a different threshold than you. And then because the whole system of digital PCR relies on proper thresholding, then it changes the interpretation of data. So think about in this scenario, how do you draw this to minimize that false positive or false negative? And same with the other plexi. And as you add more count or more targets, this picture becomes much more complicated. Okay. And one little known thing, right, is about PCR platforms right now. It's also how the instruments are designed right now. The instruments currently are designed in a way that is suboptimal for detecting multiplex signals properly. So let me show you sort of how it works, right? So say if you're multiplexing, the most typical way that we multiplex is to use different colors or different dyes. And then based on this dye, it will be this target. Then based on the another dye, it will be another target. But unfortunately, a lot of these designs of the platforms doesn't enable very clear separation of fluorophores. You can see here as an example between this green and yellow fluorophore here, they're actually quite close in the spectrum. And what you see here is even if you're trying to look at the yellow channel, you actually would see significant bleed through or a signal overlap in the yellow channel from the green dye. So it adds to that complexity of data analysis. And I know there are like ways to go about it, but you have to spend more time and more reagents to do, say, a compensation matrix. And in theory, you shouldn't need to do that. It's just a PCR reaction should be very simple. And another issue is that probes just simply cost too much and slows things down. We've all ordered TACMAN probes. You know, sometimes you order it, it could be $300 to $500 for its custom probe. So think about needing to do 4 of them for a reaction. That could be $2,000 per run. And they take up to a month to arrive. So from if you have an idea, you want to test it, you want to develop an assay quickly, this current system prohibits you to move fast. And when you think about all the optimization pieces I talked about earlier, you have to reorder probes or maybe optimize the sequence. It resets its clock. And that's why development of a multiplex assay takes such a long time. And what I'd like to talk to you guys about today is essentially a technology that Countable Labs developed. It's called Countable PCR. And the main piece that really benefits us as PCR users is that it makes multiplexing much easier without the usual trade-offs. And today I want to cover 2 main ways of how Countable PCR improves multiplexing. One is the technological aspect of things, how the technology changes how multiplexing can be done. And 2 is on the financial piece. Obviously, we don't also want you guys to be spending too much money on probes. How do you actually reduce the cost of your multiplex assay? So first of all, on the technological piece, I want to introduce you guys to like Countable PCR technology. And the most important thing, right, of Countable PCR is our ability to make millions of compartments. So for every reaction, we make 30 million compartments. So just for reference, that's over 1,500 times more than digital PCR. And depending on which platform you use, it can be more than that. And the way of generating these 30 million compartments is actually quite straightforward. It generates a way to have a bias-free amplification. So it doesn't use microfluidics. It's a simple spin column technology where you load up actually quite a bit of sample, up to 35 microliter of your DNA sample to measure, say, if you have a very diluted sample and you load everything in and everything gets captured. And through the spin column, the spin column, you put it through a centrifuge, and then after 20 minutes, what happens is that it actually spatially sort of casts these targets that you want to see into a gel matrix. So each wiggly line here represents a DNA molecule. And because we have so many compartments, it actually makes the targets you want to see be isolated by itself into this gel matrix that doesn't move, but it can still do PCR on. And then you move the sample to a thermocycler. It's a standard thermocycler that we would use. And things will amplify in situ in this tube. And when you can imagine like things are now in one of 30 million compartments, they amplify cleanly with no interference. So you can multiplex whether you do different colors, they happen in different compartments, so it's very clean. And the last aspect of countable PCR, right, is the ability to count the molecules very quickly. So we have an instrument, a benchtop instrument that allows us to load up to 96 samples at the same time and be able to image these single molecule signal in 3D. And it's probably better if I just kind of directly go into the data. You can see here I'm playing. I hope the internet is fast enough today, but you can see here, this is what we see for each PCR tube for every imaging event. So essentially, we have a PCR tube and the instrument, casts a sheet of light through this PCR tube and then images the single molecule signal that comes out that the camera sees. So essentially what I'm trying to show here is the entire PCR tube getting dissected by optics. And then you see these dots. Essentially, each dot is the signal from one target molecule. So it's a PCR reaction from one target molecule. And that allows us to just simply count. So you can see here, we just count in real time. And we just say, okay, for this sample, for these colors, this is how many number of dots we see. So it enables us to do truth quantification. So you don't need standard curves anymore. You don't need Poisson correction anymore. As a result, you get unmatched precision and low variability. So typically a percent CV of under 1% without much work. And again, you see the data is very clean and the data is automated. So there's no user bias for thresholding. So you and your colleague would get basically the same data. And I want to revisit, take some time to revisit what is the advantage of having this 30 million compartment imaging it this way. And it's really like the ability to isolate molecules very efficiently compared to qPCR, digital PCR. And I'm just throwing back the same example here, where qPCR is bulk, digital PCR is 20,000 wells per droplet or well. It doesn't really separate your molecules very cleanly. But with our platform, what you see is that since we have so many more compartments, you achieve the realm where your target molecules, I've zoomed in here, your target molecules get isolated by itself. So whether it's purple or red, they're always separate. So it's there's zero crosstalk when you multiplex in this type of system. That's also why you get things very cleanly and you actually develop assays very rapidly. It's like as if you're multiplexing, but you're actually singleplexing in reality. So that's one aspect of the technology that enables you to develop multiplex assays quickly. And the second piece is how we design the instrument. As you saw the previous slide on how clean the data looks, part of it is also how we design the instrument. So just comparing against, qPCR and digital PCR, which uses LED to illuminate your floor. In most cases, LED has a very broad spectrum, so you basically blast a much wider signal. But for countable PCR, our system is laser-based. So when you excite with a laser, say your FAM, you excite at 488 nanometer, and it just blasts that signal very targeted, and then you get very targeted excitation and detection as well. And just showing you this is what the data looks like. just this the bubble represents the signal that we get per floor in different channels. So you can see here, Countable PCR supports 4 colors. And for these floors that you are typically using, our channel discerns them very easily with minimal crosstalk. You don't need compensation matrix, and you get to actually be comfortable and have reliable signal per channel. So all in all, right, I wanted to kind of summarize sort of where the technology breakthrough is for Countable PCR. And essentially, it just makes your multiplexing super clean. And just as a reminder, right, digital PCR, it partitions, it creates different types of phenotypes that you have to fight to actually optimize. It requires a lot of deconvolution on your assay development and on your data analysis end. But for Countable PCR, just because we are able to design like the system with 30 million compartments, signal is very clean right off the bat during PCR amplification, and also our ability to detect it very cleanly. You can see here whether you're multiplexing, yeah, it's as if actually you're singleplexing. And let me show you some data on what that means in practice, right? Here is sort of a benchmarking data set where we're trying to understand sort of RPP30, which is a housekeeping gene, their expression level as we add more and more targets into the reaction. So, you know, RPP30, fairly abundant, right, with a good CT value. But what you realize is a lot of times when you add more targets, these are other genes that we're trying to amplify in the same reaction, it impacts RPP30's ability to amplify efficiently. So you can see even as soon as I add 2 reaction, it changes the CT value of RPP30 dramatically. And that is why multiplexing is so hard for qPCR, because between 1-plex and 2-plex and 3 and 4, the CT values are not the same. So how do you get actually accurate quantification in that sense? You have to work really hard to get to optimize this gap. So obviously, a lot of us use digital PCR, which does a better job, but you still need to put a lot of work into it. So this is the same, you know, assay design, RPP30, where I add more and more targets. And you can see here the count still drop, suggesting that there's still a lot of either the threshold is not drawn right, or RPP30 is not showing up very well in some of the compartments due to competition. But using Countable PCR, because everything is separated. This is, by the way, there's no optimization done. You can see that as I add more targets, the RPP30 counts stays the same. So what I like to say is, you know, I like to always think of like, now you can multiplex out of the box. And it should be that way because PCR is supposed to be a very simple technology. And now you can do that with Countable PCR. And just to highlight how small the error bars are, and that's the advantage of being able to compartmentalize into 30 million compartments. And the second part of this webinar, I wanted to introduce to you like another aspect of how to make multiplexing easier and how you can use it into your day-to-day workflow is by our Universal Multiplexing strategy that allows you to lower your cost by up to 10x. So obviously, you always want great data and also not to spend too much money on it. And a lot of us might be aware, right? Like to multiplex right now, the most default way to multiplex is to use TaqMan probes. And I'd like to introduce to you guys that TaqMan is not your only option anymore. The issue with TaqMan, while it's very powerful, it requires more sophisticated assay design. So you need to not just only order your forward and reverse primer, but you have to either design or buy off the shelf pre-designed TaqMan probes. And this probe is quite expensive because it adds 2 different conjugation and purification steps. So that gets too complicated for a simple PCR reaction. What I'd like to talk about today is essentially an alternative chemistry that we have at Countable Labs that it can be, you can try to use as a kit. It's called Universal Multiplexing. Essentially, it's a stark contrast to TaqMan probes where instead of spending hundreds of dollars for a custom probe, you don't need to do that anymore. You just need to buy your own primer pair. So if you're a SYBR Green user, imagine that it's basically the same cost as you doing SYBR Green, but now you can do it in multiplexing. And because it's a primer base sort of chemistry only, you just order desalted primers from your favorite manufacturer. Depending on where you are and where you get it from, it could be 1 or 2 or 3 days. So you can receive these orders much faster. So imagine if you have a new idea, you can do this type of work, but now with multiplexing. And to do universal multiplexing is actually quite easy. So if you're a TACMan user, you can just ditch this custom probe that you might be using, and then just add an adapter to the forward primer. And if you're a SYBR Green user, you just take your existing forward and reverse SYBR Green and you add this adapter onto it. So if you now put in multiple colors for different targets, the kit supplies 4 different universal probes and you can tack them on. And what happens is that during PCR, this adapter gets incorporated as the forward primer extends. And after PCR, you would have these adapters incorporated. And you would have these universal probes that's from the kit that's actually waiting to kind of recognize this signal. And during imaging, what happens is that you will be detecting the signal of these probes hybridizing to the amplicon that have your adapters. Essentially, it's a way to barcode your PCR products so that you can detect them in different colors. And the great thing about universal multiplexing is that you don't really have a compromise in the ability to count. So you can see here, Countable PCR supports both TACMan and universal multiplexing. And just showing you here this 4-plex assay. And actually, it's the same assay that I showed you earlier. Whether it's TACMan or universal multiplexing, you get the same quantification. And this is not without any optimization. You get the same quants and the very, very small error bars. It's thanks to the ability to do 30 million compartments and also universal multiplexing. So just a reminder what it means by having this type of easy multiplexing and low-cost multiplexing for you, right? So imagine if you're doing 96 reactions of SYBR Green of qPCR, you can quickly condense it into just 24 reactions. And for example, I used to run like 4 plates a day. And this significantly would reduce my workload. And also I get more data per reaction. And 2 is I get to lower the project cost instead of asking for $2,000 to design a 4-plex TACMan probe assay just to try it out to see if it works. Universal multiplexing, maybe just only $100, depending on where you get your primers. And third of all, get your data faster. Instead of waiting for TACMan probes all the time, now you're just, from your idea to actually getting to test the idea, could be as soon as 1 or 2 days. So in the last segment, I wanted to showcase a couple of examples of how multiplexing can make you rethink what is possible. So for those who are already multiplexing right now, you guys know the power of multiplexing. But for those who are not familiar with that, let me just give you an idea of what you could do. So this is data from our collaborator at UPenn, and Dr. Wilson has in his freezer so many like samples that monitors kidney aging, and he really wants to understand in this quick hypothesis is able to detect whether mitochondrial abundance play a role in kidney aging. It makes a great hypothesis. He's never done this type of assay before. And it is something that is very intriguing to him. And what he did was essentially he designed a 4-plex assay on universal multiplexing on Countable PCR. Essentially, how it works is that for the mitochondrial genome, design 3 targets in the mitochondrial genome plus a nuclear DNA control. And in a few days, he was able to use the samples that he has to actually accurately quantify mitochondrial counts per cell. So basically use a nuclear DNA control to normalize against these targets. And just want to emphasize this is there's no optimization here. And all 3 mitochondrial targets gave very similar results, meaning that as you increase in aging, in the beginning, as you grow up, the mitochondrial count increases, and then as you age, the mitochondrial count of in these kidney cells drop dramatically. So just want to emphasize, right? Um, Whether it's NGS or qPCR or digital PCR, it's extremely hard to capture the dynamic range, to capture this type of quantitation in such a fast and low cost manner. And the data quality is beyond what also other genomics platforms can do. And a reminder is that this type of data can be generated in less than 3 days. So whether you're writing a grant or you're trying to like answer a question very quickly, you can get to that. And for those who are in like maybe a cell and gene therapy space or in doing CRISPR analysis or doing a lot of cell culture, another customer of ours, they're really into process improvement of their cell culture sort of process. And what they were able to do was to quickly design a 3-plex universal multiplexing assay. And it's a simple proof of concept for their process improvement. This is a plasmid that they transfected, and what you can see here is he designed a target against TD tomato and a target against human DNA control, and one in the backbone of the plasmid. And what he could do in just a few days is to actually characterize this is without optimization, right? Like to be able to understand how long does it take to integrate these genes into the cells. And he thought initially it would take 4 weeks. But with this quick and dirty assay that he was able to actually accurately determine that within 2 weeks, he was able to get stable integration. And using the backbone target amplicillin resistance, he used that to understanding episomal DNA as well. And he was able to show that using his new protocol to reduce cell culture time by 50%. So not only can you use the PCR in understanding some biological question, but also for your cell culture work, you can quickly understand how you can improve your work there. And lastly, we also have a customer who is trying to develop an assay to detect cancer early. And in this application, it's really about oropharyngeal cancer. It's very rampant in some populations in LA. Currently, it uses IHC for a pathologist to assess. And a lot of times, even when you have an expert, subjectivity of the result can lead to 10% of the cases being called equivocal. So what we did is that we designed a 17-plex HPV detection assay. And this assay is actually pretty fun because it's a mixture of the very key subtypes HPV 16 and 18 that's in TACMAN. And then we also designed a universal multiplex assay in it to detect other subtypes that are not as prevalent. And this is a 4-color assay, 17-plex. And the results actually are quite cool. We've ran dozens of clinical samples and we got 100% concordance to IHC data from the pathologist. And whatever the pathologist could not call, we were able to call it with confidence. And again, this assay is only 2 hours instead of several days. So it reduces the subjectivity and also reduces the cost and time to turnaround time of these results. And I think, another way to really capture the amazingness of this platform is that think about instead of running one PCR reaction, you're actually now dividing a reaction into millions of individual reactions. And that actually changes how PCR can be done. So hopefully today I gave you an overview of how Countable PCR works and then how you can build multiplex experiments to help with your workflow. So this is the method that makes multiplexing finally easy to use so you can use it into day-to-day workflows. And two, hopefully introduce you a universal chemistry that allows you to make multiplexing assays actually affordable and fast. And three, obviously, a platform that allows you to test your ideas quickly, whether you're writing grants or whether you're writing, applications submitted to FDA. You want to be able to get to your projects and finish your projects much quicker. So with that said, a quick overview of what Countable Labs offers. It's really an end-to-end platform. It's not just about giving you an instrument where you can analyze the data, but we really believe that assays are now much truly easier to start. So we want to be able to support you from the beginning, from assay design all the way to data. So if you guys are interested, happy to work with you guys on an assay design service. But obviously, we also have different PCR setup reagents, the 30 million compartment matrix generation reagents and consumables. And then we have our benchtop centrifuge, no, benchtop instrument that enables us to do that 3D counting. And just wanted to introduce you guys, we are adding capabilities every month. And for October 2025, we will have support for 21 CFR Part 11 compliance for your lab.
Hi, I'm back. Thank you so much, Aline, for that talk. As you've been presenting, we got a lot of questions. I do want to be cognizant of everyone's time. We're about 37 after the hour. So for those who maybe only had a short period of time, I'm going to throw up another poll really quick. We will be addressing the questions that everyone's left throughout this talk. But if you got to jump or if you just want more information even after the Q&A session, please select the appropriate option on the poll. We'll get to as many of these questions as possible, but there are quite a few. So if we don't get to yours, we'll also be happy to follow up with you later. So with that, I'm just going to pull up all of these questions, scroll to the top. Okay. Eileen, how many colors are available?
Yeah, for this version, we are supporting up to 4 colors per reaction. And I like to emphasize that it's like 4 colors in a very easy manner. So you can actually develop it in 2 or 3 days. So yeah.
Here's an experiment question. Are there restrictions on amplicon length?
Yeah, great question. I didn't really have time to. There's a lot of goodies with this platform. One thing I didn't really touch base on is the ability to support really long amplicons, because a lot of times we are only kind of like we for PCR, because it's inefficient in quantification for qPCR and digital PCR. We're always saying, oh, don't design your primers to span too long on the amplicon. But for this, because everything is isolated, we've actually had data where we can count up to 5 kb. So if you're doing, I've had people who say, oh, I want to see whether I have concatenamers or whether these 2 targets are actually in the same molecule. You can study that by just amplifying a much longer amplicon. Great question.
Yeah. Thank you. So our next one. So let's go back to the workflow a little bit. How long does it take from sample preparation to reading from start to finish? We had a couple questions along the line. So maybe if you could just walk us through the steps a little bit more. Yeah.
Yeah. So for the setup part, right? It's however long that you for the PCR setup, but it will be just adding the primers and probes. The actual steps is actually quite simple. You put in the centrifuge and you spin for 20 minutes. And during that 20 minutes, 30 million compartments are generated. And then for a typical amplification protocol, it's about 60 minutes. We've done it shorter or longer, depending on the assay you're trying to do, but just say 60 minutes. And for counting, we count 1 sample at a time, but you can load up to 96. So what happens is that as you image 1 at a time, we actually spit out the data for you, within a few minutes. So like, I would say within the first 5 minutes, you'll get the sample data.
Right. And of course, that's also dependent on exactly how many targets you're probing in one tube.
Yeah, sorry. Thanks for reminding me. And that's a timing for all 4 targets. So if you're looking for 2 targets, this is cut in half, right? This imaging time.
Yeah. We had sort of another question, which maybe you can just describe the gel a little bit more, because the question is, are the compartments within a gel? So maybe just talk a little bit more on the spin step specifically and how those compartments get generated.
Yeah. Maybe I'll clarify here. So when you think about the spin column, and then when you centrifuge, using 16,000 G, it actually generates like kind of like a shower head, a molecular shower head, where you now split that 50 microliter reaction 30 million times. And then there's reagents in the bottom of the PCR too, where you think about it mixing during the centrifugation step, and that jellifies that whole compartment, the matrix. It the matrix is a kind of interesting thing when you say if you think about pet tip and poke at it, it's actually like a jello, but it's still fluid enough where PCR can occur. So the compartments already contain like your polymerase, your DNTPs, your primers and probes. And within those compartment, amplification occurs. And then you can detect that signal. Hopefully I answered your question or
Uh,
whoever's question.
um, okay, another kind of question on now on the imaging. Are the excitation and emission coupled or decoupled? For example, could you excite in green but measure in the orange channel?
Um, Currently, the excitation is coupled, I guess, in that sense, right? So if you use like FAM, it would the instrument would automatically know, okay, you've selected FAM, and then this is how we are going to do the excitation and emission detection.
Experiment question. Can you amplify RNA or perform RT-PCR?
Great question. So currently we support, you do reverse transcription outside of the compartments and then you bring them in as cDNA. However, we are, looking for beta testers for a RNA counting module that enables us to do reverse transcription directly in the compartments. So if you're interested, please let us know. Happy to chat with you to be a beta tester.
Yeah. We did have a question about 21 CFR Part 11 compliance, which Eileen sort of mentioned at the end. It came in a bit earlier, but just really do want to emphasize this will be available soon. So for those of you working on setting up GMP compliant processes, Countable PCR will be amenable to that.
And also, I would add that, right, like by being able to do automatic counting with no thresholding, it itself, that is by design, the platform is can support your GMP workflow because you don't need a technician to go, okay, this is my threshold. And there's subjectivity that adds complexity to it, right?
Yeah, and it might be worth noting on that note as well that when you actually get a data report from a Countable PCR experiment, you actually get quality metrics on every single amplification event that occurs in each tube. We call it an ID score, and it's basically a metric of how high quality the data is. So again, that adds sort of another layer of objectivity as far as kind of judging whether an event that occurred in your tube was real or some sort of, I don't know, ghost in the machine or what have you.
Yeah.
Okay, sorry, we have quite a lot of questions. I do want to stay cognizant of time. Let's do one more. There are a ton of great questions in here, though. So anybody who stayed on this long, not only do I appreciate you, but do not worry, we will reach out to those of you with names attached and make sure you get answers to your questions. So one that I think is kind of interesting for how universal multiplexing works itself. The slide showed fluorescence coming from the probe hybridization to the added tag sequence on the primers. So what prevents the probes from binding to the primers, even in the absence of target amplification?
Great question. Um, so um, Obviously this schematic is very like simplified, but I would say this sequence actually only recognize the reverse complement of the PCR amplicon. So it will only recognize it when there is PCR that has happened. So even when there's forward primer, it won't recognize it. It's basically so yeah. Hopefully that answers the question.
Yeah. One last really quick one. What is the sensitivity or LOD of your universal multiplex PCR?
Depends on your assay, right? We've gotten maybe like 0.007% VAF or MAF. It really depends on the assay and how much DNA you're loading, because VAF and MAF has a denominator. So obviously, I didn't really touch base on it, but countable PCR enables you to really load a lot of DNA. So we've done up to 3 micrograms. We've done up to like the denominator being 1 million. So depending on your assay, your sample type, you can get pretty low MAF, lower than what you would typically see in digital PCR. So if you're looking for high sensitivity applications, happy to chat with you about, like, share with you some of the data that we have with CLIA customers and how they improve sensitivity by reducing the number of reactions you would typically do by hyperwelling in by digital PCR.
Great. Okay. Again, sorry to those of you whose questions we don't have time to get to. As we wrap up, the poll's still on the screen, so if you haven't answered it but would like to, please throw up your answer now. Also, we have a really extensive resource center that includes a lot of really great resources on how our multiplexing works and how it can help improve your own lab's workflow. Actually, one of our scientists did some really great work, really showcasing the performance of universal multiplexing, its flexibility, and just how well it works no matter, like Eileen said, how many targets you're looking at, whether it's high or low or anything in between. So check out the tech note. You should see a little bubble popping up on the bottom of your screen for that right now. And with that, I really want to thank all of you for joining us and sticking around for the whole presentation. I hope everybody's learned a little bit about how Countable PCR and Universal Multiplexing really made multiplexing actually for real easy and approachable.
Thank you. Thank you very much.
Everyone.